Hello everyone. Today we're going to explore the concept of Fisher information and see how it provides a powerful framework for understanding and optimizing physical experiments. We begin with the fundamental challenge of all experimental science. In nature, there exists some true, unknown value that we want to measure. We'll call this parameter theta. Our measurement process, however, is never perfect. It's probabilistic. This means that for a given true value of theta, our measurements, which we'll call x, are scattered around the true value according to some probability distribution. In a real experiment, we don't see the underlying distribution. We only get a handful of these measurements, which are scattered around the true value that we are trying to find. So, this is our core task. We have a set of measurements, x, and our goal is to use them to make the best possible guess about the true unknown value of theta that produced them. Crucially, there is a fundamental limit to how good our estimate can be. No matter how clever our analysis is, the uncertainty in our final estimate of theta, its variance, can never be smaller than a certain value. This limit, on precision, is known as the Cramér-Rao Lower Bound, and it is defined by the inverse of a quantity called the Fisher information, I of theta. More information means a lower possible error. So how do we get from our data to an estimate? We use the concept of likelihood. Instead of asking, given the true theta, what data would I get? We flip the question around. We start with a single data point that we actually observed. Then we ask, given this specific data point, how likely is every possible value of theta? This defines the likelihood function. For each hypothetical value of theta on the x-axis, we calculate the probability that it would have produced our observation. This creates a curve. The most intuitive guess for the true value of theta is the one that makes our observed data most likely. This corresponds to the very peak of the likelihood function. This peak is our best estimate, known as the maximum likelihood estimate, or MLE. Working with likelihoods, which often involves multiplying many small numbers, can be inconvenient. It's much easier to work with a logarithm. The log likelihood function has its peak in the same place as the original, but its shape is what's truly important. And here we arrive at the formal definition of Fisher information. It is a negative expected value of the second derivative of the log likelihood function. In simpler terms, it is a measure of the curvature, or the sharpness, of the log likelihood peak. Let's compare two scenarios. The red curve represents a high information experiment. It has a very sharp peak. The blue dashed curve represents a low information experiment, with a much broader peak. For the same single observation, the high information experiment gives us a much more certain estimate of theta. We can also think of this in terms of the slope of the log likelihood, which is called the scores. For the high information case, the slope changes very rapidly as we move away from the peak. The function is very sensitive to changes in theta. For the low information case, the slope is much flatter, the function is less sensitive. The rate at which this slope changes is the curvature, and that curvature is the Fisher information. Let's make this more concrete by comparing the likelihoods from two different experiments. On the left, we have a precise measurement characterized by a small uncertainty, or sigma. This gives a lot of information. On the right, we have an imprecise measurement with a large sigma, which contains little information. For the precise measurement, the Fisher information, which for a Gaussian is proportional to 1 over sigma squared, is large. For the imprecise measurement, the Fisher information is small. The story this tells is crucial. A single data point from the precise experiment on the left tells you a lot about where the true theta could be. It tightly constrains the possibilities. The exact same data point, if it came from the imprecise experiment on the right, is much less informative. The true value of theta could be almost anywhere within that wide range. The value of data is inseparable from the quality of the experiment that produced it. So, why do we bother taking more data? Let's see what happens as we accumulate measurements. We start with one measurement, which gives us a broad likelihood function. Now, we add a second measurement. Since the measurements are independent, the total likelihood is the product of the individual likelihoods. Notice that the new combined likelihood, shown in red, is already sharper than either of the individual ones. As we add a third, a fourth, a fifth, and a sixth measurement, the same thing happens each time. Each new piece of information helps to narrow down the possibilities. With all 6 measurements combined, the resulting likelihood function is dramatically sharper than any single one. This is the magic of data aggregation. Mathematically, multiplying likelihoods is the same as adding their logarithms. This is why, for independent measurements, the total Fisher information is simply the sum of the information from each measurement. Information accumulates. Let's now simulate one full estimation trial. We'll compare an experiment with only 2 samples on the left to one with 10 samples on the right. We see the true underlying distribution as a dotted line, and the random samples we draw from it. Now, from these samples we calculate the likelihood function and our best estimate, the MLE. For the case with only 2 samples, the likelihood is broad and the estimate is quite far from the true value. For the case with 10 samples, the likelihood is much sharper, and the estimate is much closer to the truth. We can explicitly mark the estimation error for this single trial. On the left, the error is large. On the right, it's much smaller. This single trial illustrates how more samples lead to more information, which leads to a more precise estimate. Of course, a single trial could be lucky or unlucky. To truly understand the quality of our estimator, we need to repeat the experiment thousands of times and look at the distribution of the results. On the left, with only 2 samples per trial, the histogram of our estimates is very wide and spread out. The observed variance is large. On the right, with 10 samples per trial, the estimates are tightly clustered around the true value. The conclusion is clear. With more information, our estimates are far more precise and reliably cluster around the true value. This brings us to the grand finale. The Cramér-Rao Lower Bound, which we calculate purely from the Fisher information, gives us the theoretical best-case variance for our estimator. In this plot, the calculated lower bound value for each case is shown. As we can see, the observed variance of our estimates almost perfectly matches this theoretical limit. This means our estimator is efficient. It extracts all possible information from the data, achieving the fundamental limit of precision. This is not just a theoretical curiosity, it's a powerful tool for experimental design. We can plot the best possible precision we can achieve as a function of some experimental resource. In this case, the total number of photons we collect in a physics experiment. To get a lower error, we need to move to the right, which means collecting more photons. We can frame this as a choice. Experiment A uses only 10 photons. The theory tells us our relative error will be high, around 32%. This may be too imprecise for our needs. But what if we could do even better? This is where the story gets really interesting. Imagine we have a fixed budget of photons, say 80 photons, as marked by the dashed line. The standard approach gives us the precision on the magenta curve. But is it possible to beat this limit? The green curve suggests yes. By controlling other, more subtle degrees of freedom in our experiment, in this case by shaping the wavefront of the light we use, we might achieve a much higher precision for the exact same number of photons. This final slide illustrates the concept. The standard experiment uses a simple plane wave of light. For a given photon budget, it gives us imprecise estimations, like a scattered group of arrows on a target. The advanced experiment, however, uses a carefully shaped wave of light. This wave is specifically designed to interact with the unknown parameter in the most informative way possible. For the exact same number of photons, this method can yield far more precise, optimal estimations, like a tight grouping of arrows in the bullseye. This is the frontier of modern experimental design, maximizing Fisher information, not just by collecting more data, but by collecting smarter data. Thank you.